Okay, so let me check. Dr. Sosa is, yes, Dr. Sosa is available. And it is my pleasure to introduce today's special guest speaker, Dr. Glauco Souza. And as you see in the chat box, and a number of people already asked a question, and then some of the special population we isolate from this kind of the 3D differentiate tissue, and then how we can do it with a three-dimensional structure model. Yeah, that is the bioprinting. So Dr. Sosa is the director of global application development bioscience at Greiner Bio-One. And he brings over 20 years of the experience in life science innovations and spanning the translational research, technology development, and also commercialization. So Dr. Sosa is widely recognized for the pioneering magnetic 3D cell culture and bioprinting technology, integrating nanotechnology with a high throughput biological system. Actually, our center and some official automation team, and he is actively collaborating on this. And his work has significantly advanced the development of the physiologically relevant 3D tissue models for research and also drug discovery. And he holds research into broadly adaptable platform. And then also, prior to his current role, he served as CEO and Chief Scientific Officer of Nano 3D Bioscience, where they successfully secured funding and also built the strategic partnership with leading institutions such as Scripps and also Cold Spring Harbor Laboratory. And his research contributes, including high-impact publication in journals such as Nature Nanotechnology, PNAS, and multiple scientific journals, and particularly in the area of the 3D tissue culture using magnetic gravitation system. So today, Dr. Sosa will present on 3D biology to the future, discussing emerging direction in 3D system and their application in scalable and translational research. Please join me in welcoming Dr. Glauco Souza.
Thank you very much, Kite. Very nice introduction. You're very generous. But so we're gonna talk about 3D cell culture, and we're gonna talk mostly about magnetic 3D cell culture, which we divided into levitation and bioprinting. But first, I'm also this. We all in academia like to publish, right? And I like to point to this because I'm one of the editor for SLS Discovery. It's a good journal, impact factor of around 4. So, and we had a 3D biology protocol before, which was very successful, was a special issue. But now we're coming with a special issue for negative results. So, and that's one of the things too, that it's very fitting for our field of 3D biology. Because one of the aspects, especially when you go to drug discovery, high throughput screening, one of the motivations today, and I'll show some data here, some results, for using 3D cell cultures is to actually reduce the number of false positives, right? So, in the negative results, it's almost like put our money where our mouth is, because everybody says, I wish somebody would have published their negative results. So this is going to be a great opportunity. The special issue, it's probably going to come before the end of the year, but I'll let you know. I just want to let you know, since we all like to publish, especially the stuff that we have in our drawers. And again, one more thing, negative results doesn't mean bad results. Negative results means a hypothesis that we tested and they didn't work. The experiment's great. But all right. So 3D cell culture, right? There's various aspects from 3D cell culture. As Kite mentions, I've been in the field for 20 years. I started Nano 3D Biosciences. And back then, there was a lot of hand waving. Is 3D biology really liking human in vivo biology? Is it really different from monolayer cultures? So, but April 10th of last year, I think it's a very relevant date for us because the Modernization Act now allows submission of data without animal studies for a category of drugs, right? So. So now that we don't have to use animals, I'm not saying that we're going to stop using animals, but 3D models, NEMS models, which is a new modalities applications, will be accepted by FDA. People are going to use them more because you can use human cells, and and it's much less expensive than animal studies. So, the origin of magnetic 3D cell culture. The technology came from a collaboration between MD Anderson and Rice University. I was at MD Anderson first. I have two friends from Rice University. I was an Odyssey scholar, and then we developed magnetic 3D cell culture, and we spun off a company called Nano 3D Biosciences. I ran the company for 10 years, and in 2018, Greiner acquired the technology. So that's why I'm Greiner now, and my goal is always to develop applications with magnetic 3D cell culture and without magnetic 3D cell culture. But one quote that I think is very applicable for our world of transitioning from 2D cell culture to 3D cell culture is everything should be made as simple as possible, but no simpler, right? 2D cell culture is fairly simple, right? You put in on a plastic, they stick, and you can do a lot of things with it. You go to 3D cell culture, you're adding complexity. It is going to be more complicated. There is no question. You're going to require to optimize. But sometimes we over-engineer. We make it more complicated than it has to be. So, we like to make it simple, but there is a limitation for how simple we can do things. But one thing that you appreciate when you go from the world of 2D cell culture to 3D cell culture, and Omar kind of touched on many of these things, is that you really start appreciating how valuable it is cells stuck to plastic. Why is that? Because you want to change media, which is a basic foundation of cell culture. Your cells are stuck to plastic. You can remove the media and then add the media. You want to do immunofluorescence, staining, downstream manipulation. The cells are attached. It's very easy to handle them. But when you lose the attachment, when you go to 3D cell culture, that's not easy. Changing media, it's not simple anymore. So it is added complexity when you go to 3D cell culture. Now, what we did to make it simpler is we came up with these nanoparticles that it can stick to the cells electrostatically. It's a nanoparticle assembly of gold, iron oxide, and polylysine. It just sticks to the cell, non-specifically, just electrostatic. And once you do that, we can use magnets to bring the cells together. As Omar mentioned, cell-cell interaction is key for 3D biology and in biology in general. So instead of using gravity, as people use with round bottom or hanging drops, we just use the magnetic field to bring the cells together. And we can do it with the levitation when we put the magnet on top, and that's where we started. And you can put it on bottom. We started the levitation, the magnet on top, because when we started, there was no low attachment surface, ultra low attachment surface. There was no cell repellent. So we wanted to get it away from the surface. So we put the magnets on top for cells to lift up and form the 3D structures. So now the ultra low attachment surface came about. Now we can use also the bottom to really print and position and miniaturize the cultures very precisely with magnetic fields. And again, we have different kits, different. This is the levitation system. This is the bioprinting system. And I have a video here, a cartoon that shows exactly how magnetic 3D cell culture works. And one thing though that you may be thinking, it's on the video. Once the cells come together with the magnetic field, you can remove the magnet. It's all cell-cell interaction that makes the 3D assembly. So you do not need to leave the cells on the magnet. So here is a quick cartoon that shows the workflow. So these are kits. You have your plate, your magnet, different throughputs. You have your nanoparticle vial. And then first thing you do, you add the nanoshuttle to your cells. That's simple. No special media. You just add to your cells. If you're having monolayer, the nanoparticle will just settle on. And next day, when you wash them, you wash the free nanoparticles. A few nanoparticles will tag your cells. You trypsinize, you do whatever the process for detaching the cells you need. And then you count the same way you do. And then when you collect your cells, you put the cell repellent, the ultra low attachment plate on the magnet drive. So there's a magnet right in the middle of the magnet. And as soon as they hit the bottom of the well, they'll come together. And remember, this is flat bottom. So this is a little mix just for all the cells to kind of move around, like 5 seconds. 15 minutes on the magnet. Again, you leave it on 15 minutes on the magnets, that's it. And then you can take it out and your structure forms. Of course, the assembly is over time, but the cells come together in the middle. Now you want to change your media when you want to add your drug, fresh media. You put it up on the drive that has one magnet between the 4 wells, and then it moves the spheroids to the side. Now you can very easily pipette the cells out. So we have in different formats. And one thing that is important too in 3D cell culture is scalability. A lot of times you're working on, like for example, if you're using round bottom 9.6 well to 1.536, if you want to go to lower throughput, you don't have it. You have to change your technique. So the magnetic 3D cell culture is truly scalable. You can go from 6 all the way to 1.536. So basically, it's a magnet in the middle of the well. So you put your plate, the cells come to the magnet because they're magnetized. You take the magnet out after 15 minutes, and the cells assemble in a 3D structure. So I like to show this for a few different reasons. One is because as a small company, Nano 3D, before we became Greiner, sorry, I'm on some auto here, but I'll keep playing. So just to show, we have a lot of high impact publications, very good publications. And the reason we show that is because we use nanoparticles. And we've shown over and over the nanoparticles are bioinert. They don't do anything. We have, we compiled a 5-page white paper that would show nanoparticle with nanoparticle, without nanoparticle, with viability, proliferation, stem cell differentiation, inflammatory stress, inflammatory oxidative stress, gene expression, drug sensitivity, and there's no difference between having the nanoparticles or having the nanoparticles. Again, it's the concentration that makes a difference. And cells are very buoyant. You only need a little bit of nanoparticles to make it work. So these are just different results we have that touches in different aspects of benefits of using magnetic 3D cell culture. One of them is, you bring your cells together, you can generate these spheroids really fast. You can do co-culture. Here we publish in Nature Nanotech. It's a co-culture of glioblastoma and normal human astrocytes that you culture them separately, magnetically brought them together, and you can see the glioblastoma invade the normal human astrocyte. The bottom right is one of my favorite image, a confocal image of a adiposphere. Red here is pure lipin, adipocyte marker. The green are endothelial cells, GFP labeled endothelial cells. And this is a confocal image that we really only were able to get this beautiful image because we used the magnet to hold the spheroids down and be able to wash and stain the way we do in 2D, basically, to facilitate this process. And of course, since Omar saying this is our, we also have worked with a lot of iPSC cardiomyocytes. And again, as Omar said, it's not AI. So you can see the cells beating. We took from the cardiomyocytes, we labeled them, and we just bring them together. And very nicely, they just have a beautiful phenotype of the heart cells beating as they're supposed to, right? And also independently, AstraZeneca, which also paired with some of the results Omar showed, they actually created a 384 screening. As Kate mentioned, our tool is very scalable. You can go from 6 all the way to 1536. In this case, they use a 384 384 electrode plate from Nanion. And what they did, because the cells are magnetized, now we didn't have to break the spheroids and disperse the cells. We can take the spheroid and localize right on top of the electrode. And it gave beautiful electrophysiology signal with the cells. And also, as Omar mentioned, they also co-culture the iPSC cardiomyocytes with fibroblasts and definitely improve their results. So this is published on SLS Discovery. And if you guys have any questions, feel free to reach out. I'll be more than happy to touch on it. Another tool that we came up with, one thing that is, I keep saying, changing media, it's always complicated. Immunofluorescence is always not trivial with a 3D cell culture because the cells are not attached. So we came up with, we call the MagPen. It's a pretty neat tool to transfer, make basically full media exchange. So, and this is a cartoon that shows exactly how this works. So you magnetize your cells, you make your 3D spheroid, and now you want to put the plates in a fresh media, or you want to do immunofluorescence staining. So it's a kit, and you have a magnet array, 24 and 96, you have a sleeve, and you have your ultra low attachment plate. So remember, you have your magnetized cells on the bottom, right? So now I want to transfer them. Like a pipette, people cut the end of the pipettes to transfer the spheroids. We put this sleeve inside, okay? It's sterile and cell repellent. And then we put the magnet. What happens is the spheroid is going to go to the magnet, but the sleeve is going to block them to touch the magnet, right? So there's a separation there. But now you can pick up your spheroids and transfer to a fresh plate. So you put it in a fresh plate, and now actually you put the magnet on the bottom just to gently pull them from there's some, low adhesion and surface tension between the plastic and the cells. You remove the magnet, and then you put the magnet on the bottom, and the cells gently come off and that's it. And here we have 24, 96. We also have a single. So now you take the sleeve out, and you take the magnet out off the magnet. You take the plate off the magnet, and that's it. So, and you can continue your culture. If you're doing immunofluorescence, you can do like washes by just using like you do with the paraffin embedded cassettes when you have to do the xylene washes. You can basically use that kind of workflow. So it's an enabling tool. The magnetic 3D cell culture, no matter what technique you're using, it will make it easier to manipulate the cells. And the analogy I like to use, who hates dropping paper clips everywhere, right? When it drops, you always cuss it, right? Now, if you have a magnet, it's very easy to pick it up. It's sort of the same idea. It's just easy to pick things up. So as Kite mentioned, we have we had a large grant from an NCI with Cold Spring Harbor and Scripps back in when we were in Nano3D. And this grant was to validate our high throughput screening platform, the 1536. And also at the time, we still had questions. Does 3D is different from 2D? And one of the hypotheses we have, does 3D cell culture give less false positives than 2D cell culture? So we compared both side by side with in different configurations. And here we use pancreatic cancer cells that was isolated by the group of David Tufson and Cold Spring Harbor. And we were able to regenerate, single spheroids per well in this case, shown here, a full 1,536 plate. And one thing that we can do too, from a practical point of view, when you're using a flat bottom plate, right, which is ideal for imaging, right? If you have a round bottom or a thin drop, there's a lot of limitations in imaging. The round bottom becomes a lens. So if the spheroids sit a little bit to the side, you get kind of all kinds of optical aberrations. So you wanted the spheroid sitting right at the bottom, and that's not trivial. On the hanging bottom, the hanging drop, you have sort of the same problem too, but now the problem is the traveling distance. For to get high resolution, it's not trivial. So in this case, so we have flat bottoms. When you have cells on a flat bottom in 3D, they tend to move to the side. And when they, as you see here, this is a 1,536 plate where some of the spheroids move to the side. Every dot here is a spheroid. So if you look at here, you lost about a quarter of your data because the spheroids move to the side, and the microscope won't find them at the side of the well. So what Scripps was doing in their workflow, before going to the imager, they put it on the magnet for 30 seconds, and the spheroids always center. So you don't lose anything. Very easily, you can just center them, and now the microscope know exactly where the spheroids are. And this works also with organoids. And as I'm going to show here, we can work with spheroids, organoids, and this is some of the results we showed. And in the way I like to again, I usually have a disclaimer, and I didn't do it here. I'm not a biologist. I'm a physical chemist by training. So I build tools, and the way I think of cells over the years is that if you bring them together under the right conditions, they do what they're supposed to do. So in this case, we have HT29 ovarian cancer cell line, PANC1 pancreatic cancer cell line. We use the magnet to bring them together, and you can see over time, 72 hours, beautiful spheroids. That's great. Now, in this work, we actually took patient cells. In Cold Spring Harbor, separated the cancer cells from the cancer-associated fibroblasts, okay? And we tested them separately. So AGM1 is one patient, HT1 is another patient, and the AGM1 calf is the pair of cancer-associated fibroblasts separated from the tumor cells. The same with the HT1. So when we take the calves, we magnetize them, bring them together, they make beautiful spheroids. Fibroblasts usually make very nice structures spontaneously. Now, the cancer cells, we bring them together, and no matter what, they will not come together into a single structure. They make organoids, and we characterize. They have the lumen, they have the same characteristic of organoids, but they still magnetize. We can still center them and manipulate them. So the results here. So remember, one of the hypotheses we have was that 3D cell culture will give you less false positives. So in this work, to test this hypothesis, we used the FDA compound library without the cancer drugs, okay? So it's about 3,000 compounds, and we tested in 2D, 3D, and in 2D with the patient cells. In every system that we tested, 3D always gave less false positives, which means more cells killed compounds in 2D than in 3D. Now, how do we know it's a false positive? It's a very tough hypothesis to test in a way, especially high throughput. So we went with the FDA library that doesn't have drugs prescribed for cancer. So the assumption we're making here is that if it's killing the cells is a false positive because it wasn't supposed to kill cancer cells. Okay. So one thing we did, a step further we actually plotted the inhibition level for the drugs in 2D versus 3D. So 2D on the x-axis, 3D on the y-axis. And every dot here is basically the IC50 for each, or the drugs that were below the IC50. So white quadrant cells basically didn't die. The red, the cells died only in 2D. The green quadrant, the cells died in both 2D and 3D. But what's very interesting, and it was that basically the only drug that was really active in 3D was disulfiram, and it was in clinical trials at the time. Disulfiram is a nasty drug for drug addiction. And again, I make the joke, I was just happy that was an aspirin. But yes, we did pick a drug that was only active in 3D. The compound did not kill the drugs when it was in 2D. And we went beyond, we did 150,000 compound screening, again, 2D versus 3D. And if you're interested in drug screening, one number that is very important is the Z prime. Basically is, the number of standard deviations between your positive and negative control. And we've have, anything that is Z prime above 0.5 is considered excellent. So repeatedly, we are having very good Z primes with our studies. Next, one thing also, automation. When you go to automation, when you change media, and it's not trivial when you're using 3D cell culture. And here we show a quick movie that, you can put your magnet, take your magnetized cells, put your flat bottom there. This was done with TCAN a few years ago. So you can use your pipette, your magnetized cells, you dispense into your 384 plate, the robot puts the plate, they use 60 minutes on the magnet, and then it goes in the incubator, and that's it. You want to change media, you put it back on the magnet, you take your media out, add media, and that's pretty much it. Very straightforward. We also have, this is a really cool publication that we published in Biomaterials, where we actually took human dental pulp stem cells from the patient, and we differentiate into what we call mini salivary glands. And we also actually were able to re-innervate the tissue, and we show the beautiful re-innervation of the tissue in vitro. We also have worked with pancreatic islets for production of insulin. And again, using the magnet, it makes it much easier to do the co-culture. We can bring different cell types together. We can bring one magnetized cells, the other one that is not. And also to remove supernatant. It's very easy because you can put the magnet on the bottom and just aspirate or pipette out the media. So this is a pretty cool publication. People usually ask, why you call 3D bioprinting, right? Magnetic 3D bioprinting. So we can bring the cells into a dot, but we can make it more complicated. We can use magnets to pattern the cells in different places. So these are GFP labeled glioblastoma that we can, create more complex structures. One of my favorite work when we're in Neuro 3D, we have a cases grant. We actually send cells to space, right? So it's a fair question. Why is cases spend money sending cells to space when there's no gravity? Cells grow in 3D cell culture without gravity, right? And one of the answers is it's astronaut time is very expensive. It's very complicated to change media of cells in space. So here we send the magnetized cells, we can put the magnet on, and now they can just suck the media out and suck the media in. And another aspect too of this, which people forget, when you're planning a project to send to the space station, any gram of material costs money because you need fuel, lots of fuel to bring to the space station. And the way they change media with cells is by exchange. So they always have an excess volume of whatever fixative you have or media in that exchange. And that's very expensive. So this way you can really just use very simple and just normal amount of medias to do the media exchange. And one more thing, I wanted to be the first one to grow a monolayer in space. And cells don't survive. You can imagine without the vibration, there's no monolayer that survives the way to space. So we send cryopreserved. And we wanted to put the cells on a membrane and see if they grow flat and spread. And we were able to stick the cells to the membrane using magnets, but they did not spread. They just kind of grew outwards, stuck to the membrane, but they did not really, we were not able. I have a few ideas for new experiments, but we have to go back to the station. In the next few years, we probably will. So I speak fast. I went through a bunch of things here, and again, I had I had a big mix up, and you guys didn't see it, but Taste saw. I thought it was I had one calendar that said my presentation is at 11, another one that was at 12. So I was like, oh no. But everything worked out. You guys never heard, but again, this happens to everybody too. So all kinds of technical issues can happen. And we have collaborated with lots of people. We have had NIH funding, phase one, phase two SBIRs from NIH, from NSF. We have DoD, cases funding, state of Texas. And we had lots of collaborations. And I can answer any questions. If anybody interested in learning more results, I have a lot more results. Feel free to let me know.
Okay, good. Thank you very much for excellent, very interesting presentation. And also, just raise the hand to directly ask a question, or just leave your question on the chat box while you are presenting. And I have one question. Since single cell stage in multiple different cell type, for example, primary stem cell or differentiated cell out of the stem cell, and also primary cell, even cancer cell is highly vulnerable to cell death. They stay as a single cell. And the definitely magnetic nanoparticle-based system may reduce time spent as a single cell suspension. Do you have or introduce any data comparing some kind of organoid formation with without magnetic nanoparticle?
Yes, actually all the study that we did with scripts too, we did with and without nanoparticle on round bottom, and it correlated the same. So we did a lot of studies. Now, one thing though that actually improved the output of the technique, it's a +1 paper that we actually took a PDX from triple negative inflammatory breast cancer, okay? And we compared the PDX to the 3D culture, the magnetized culture. Exact same phenotype. The markers distribution is the same, but two things. Those cells, they would not grow into D, which is why we did not have in the paper a 2D comparison, one. Two, we did not have a round bottom on other techniques comparison because they will also not grow. You needed to push them together, improve the cell-cell contact with the magnet for them to actually show the right results. And we do have lots of work, and we have a 5-page that show stem cell differentiation with and without nanoparticles, no difference.
Okay, Omar, turn on the mic.
That was sorry, yeah, that was I was on mute. I couldn't get that was super interesting. Thank you, Glauco. This is really, really cool. I have a couple questions for you. I'm seeing some questions also come in the chat. In terms of you mentioned that there seems to be quite comparable like 2D and 3D outcomes. Is this done with
not that comparable, I would say, but that's okay. Go ahead.
Sorry, I meant in terms of like the phenotype of the starting cell population. Like you're not really changing the original blood. But have you tested that with like varying magnetic field strengths, like the strength of the magnet? Could that be even a downstream approach where you can increase or decrease the magnetic field, or like kind of the push or the pull there?
Yes. So there's different ways we can control the magnet field, usually with distance, because we don't actually the development started with electromagnets. That's how we made a chip, and that's a story for, it's a long story. It's a fun story. But with magnets, two things. one reason for us to, and we have shown in papers with several papers, the time on the magnet, we leave only 15 minutes, right? So, and then you take it out. You only put the magnet on to manipulate them, with exception of, for example, the PDX system that only worked with the magnet. And that is really, it's cadherins. I think it's just cell-cell interaction. The cells are stressed because you're lysing them, you're manipulating them. So when you push them together, they do much better. That's one thing. It's very cell contact mediated. And we have more data too. I didn't have time to show. Now you can, and one thing though, you've done cardiomyocyte cultures. When they start beating the first time, we're all like fascinated. You think like you have a buddy in the room, you keep going check to see they're beating, how it is. So one time we did the culture, and I was always the first one in the lab, in the office, and I would go look at the cardiomyocytes. And one time I came back, one of the batches we tested, we looked at it, they stopped beating. And I'm like, oh no, which can be good or bad, it's so just for the sake of curiosity, I told I put it back on the magnet. Let's leave it on the magnet for a few hours and take it off and see what happens. They start beating again.
Oh, wow. Oh,
which,
wow.
again, you're
So
squeezing
cool.
them together, which makes sense. If you think about it, you just push them, and then they start beating again. So
cool.
yes. So, and if you have mechanosensitive cells, they're probably going to be more susceptible. But we try to minimize the amount of the magnetic field. On the levitation, it's a long distance, so it's very weak force that you put it on the magnet. So yeah, but that's one thing that yes, you can manipulate if you need to, and you can get bigger magnets or small.
And then, can you also add the the nanoparticles? Can you add them to an already formed structure,
Yes.
or do you need
Yes.
to? Okay, that's pretty cool.
Yeah, we have a protocol to that, and we have a couple papers with protein fluidics where they have a microfluidic system, and they want to position exactly in this correct spot. Even with seahorse from Agilent, you can use that to guide them to the electrode.
Super cool. Super
Yeah.
cool.
Yeah.
I
Thank you
see
very
some
much.
questions in the chat for you.
Yeah, I
Oh,
will
sorry, go ahead,
look
Keith.
through.
Yeah.
I will look through. And then just continue to raise the hand to ask a question. And first, something related to Omar's question. And then from Chu, and do magnetized cell culture affect their differentiation potential? Or are there concerns that new user may not be aware of?
Again, we usually do the controls, which is fairly strict. On the stem cells that we've done, we actually published one with the carbon monoxides. And we see the same kind of cavities that you show there with the cultures. We haven't seen any difference between the magnet or no magnet. But if you have a mechanosensitive cells, you could. But that's why we try to leave it on the magnet not very long. And again, Omar understands that when you have a system that you sell, if there's something that doesn't work, you're the first one to say it doesn't work or does something, because I usually joke that we publish less than 5% of what we do, because most stuff we do doesn't work. I know what doesn't work, and we haven't seen one case that it's a problem.
Yeah. Next question is from Jeanette. And have you explored functionalized magnetic nanoparticle beyond the simple assembly, such as bioactive or ECM, mimetic coating? Or is there a need for this to reduce the necrosis, toxicity, etc.?
There's different reasons. We actually have explored doing creating a magnetized ECM, right, using decellularization. It works really well because the nanoparticles end up in the extracellular matrix. So you can use the magnet down to hold. If you're familiar with decellularization, organ decellularization, right? They basically take a heart, they plug some tubes in it and flush with detergent, right? And then you end up with the extracellular matrix. So we've done that with, we have fibroblast cultures. We put a magnet on and wash, and you end up with basically the magnetic now coated with the ECM of the same cell. So that's one of the things. Functionalizing, again, people ask that. For the sake of simplicity, we like to use just the promiscuous nanoparticle that it sticks to all cells. It's just electrostatic, it's polylysine, same tricks you do with but you could, in principle, you could modify the nanoparticles to functionalize the nanoparticles, yes.
Yeah. Next question is
Hey, Itai.
yeah.
Sorry, I have a question that's maybe related to that,
Oh,
if you
yeah,
don't
sure.
mind. Yeah.
Go ahead.
Glauco, you just raised up a really cool point. Have you guys ever tested, organoids that are formed because the nanoparticles remain there, right? Like they're inert, but they remain.
Yeah.
Have you tested whether they can be used in order to cryopreserve and thaw out those organoids intact if you can maybe manipulate the way they, distribute heat across the organoid?
We haven't. Okay, simple answer. We have magnetized cells and cryopreserved, and it works beautifully. That's how we send the cells to space. They were just magnetizing on the ground, and they just thawed out and grew them. It worked beautifully. So that's a good example. We have not worked very hard in cryopreserving organoids. But it's tricky. But yeah, maybe even for the sake of manipulation, removing them, but we haven't done much.
Yeah, it's gonna be really
If you're
interesting.
interested, I'll be more than happy to. We can talk about it. Yeah.
I think we should we'll
Yeah.
connect. That's really
Yeah.
interesting.
Yep.
Yeah, next question is about comparison between scaffold-based engineering or even bioprinting. There is a sophisticated special bioprinting versus your bioprinting is more focusing on the mass production. So question is that where do you see the biggest bottleneck today in scalable organoid manufacturing matrix material, reproducibility, or automation? Do you see magnetization technology actually replacing scaffold-based engineering?
That's a big question. And again, I don't think as the magnetic 3D cell culture as so much as replacing, but enabling to do more complex handling. I'll give an example. You're using matrigel, right? You want to culture in a 96 well. You have your matrigel, and you have very few number of cells. You put it on your well, right? It's not trivial to go find the cells and visualize them, right? Because you have to scan the bottom. It's not trivial. Now, if you put them on the magnet where you dispense your cells, and the cells are magnetized, you localize all the cells on the matrigel in the middle. So from a practical point of view, it's much easier for you to find, right? We have magnetized spheroids put on matrigel to look at the sprouting. Again, you can localize because magnetic field put them in the exactly place. Just like holding on the electrode, that's not trivial, as Omar probably knows. It's not trivial. And it was pretty neat the way they did, which is probably similar to guys you did. They coated the electrodes, AstraZeneca, with fibronectin. But now the spheroids, they can put the magnet right under, and they go sit right on top of the electrode, and they sit there. Very reproducibility. So I think it's more of an enabling tool to make it whatever technique you're using easier.
Okay, good. Any other question? Raise a hand, final minute. Yeah, actually, our center is collaborating with the Thermo Fisher Scientific automation team and also Dr. Souza to build a 3D sarcoma lung metastasis model. So incorporating patient-derived tumor, and then also we coated with the endothelialized vessel-like structure. And then iPSC-derived lung tissues around the body and then autologous dendritic cell and the tumor infiltrating lymphocyte in 384 well format. So therefore, what? So Dr. Souza's tool is really best for the scalability. And then sophisticated bioprinting and then scalable this bioprinting is not just a competition, it's complementation. Depending on what is the goal, we can use one to the other. In our case, we love scalability. So therefore, 384 well bioprinting with multiple different tissue type, including immune tissue, perfect to mimicking the metastasized sarcoma in lung. And if we make 10 of these plate, how many sample number? 3,840 conditions. And then this one using the robotic automation and then also automated high contents confocal imaging using the AI-based automated imaging analysis of the 840 samples. Can we do with our eye? No. So this one is need robotic automation. So therefore, if you are interested in this type of the experiment, please contact us and then we can just start from the grant writing. And this AI-used stem cell study will also be presented by another special guest by Dr. Christina Correa next week. Dr. Glauco
One
Souza?
more quick question. One quick
Yeah,
note on the last question. Just after you start, you kind of answered the question too. I just want to complete that. One key difference between, for example, the extrusion type of tools and the magnetic bioprinting, right? They're different. It's time of extrusion versus time of printing with the magnet. Again, you dispense the cells, you put it in a magnet. It's immediate. It's a few seconds. You have your cells coming together. With extrusion, it takes a lot of time for you to extrude all the wells, and also the throughput, the miniaturization. You cannot go on 1536, as I know, I think. It's not trivial. And the time of dispensing on a 1536, it's very long when the cells do not like to sit on the nozzle very well. So there's people are doing better and better in this, but there is some practical and limitations on that when you compare the two. If their cells are sensitive, you gotta look careful which technique you're gonna use.
Yeah, I totally agree because even if you isolate specific cell type from 3D culture, and once they become a single cell, they are really, really vulnerable.
Yeah.
And then how fast we can form cell-to-cell contact is also major factors to control your outcome of the outcome.
We think about it, but even media conditioning, right? And there's cells that play it on one layer. Okay, if you put too few, they know where they are. You know, you need they don't like to be very dispersed. So there's a lot of things that promoting cell-cell interaction is key.
Yeah, definitely. This is the really good conjunction, something like a computational biology, AI-based image, automated image analysis, data collection, immunological, some cell movement, and then also immunology, cancer biology, stem cell biology, more different technology and idea and the area combined together, your research will become much more stronger.
Yep.
And any other things, Omar, Tom, and then Sosa want to mention? No?
Thank you very much for having us. Yes.
Okay.
Pleasure to see you, Omar. Great talk too.
Yeah, thank
Thank
you.
you as well, Glauco.
Yeah, thank you, Omar, Tom, and Dr. Sosa for excellent and insightful lecture. As discussed, while some experimental example is based on the Thermo Fisher platform or Bio-One Greiner platform, and then the principle of this, all of the technology, you can broadly applicate to the other tools and the technology. Just this kind of lecture will give you the baseline. So therefore, do not much complain about why we are talking about one company or the other company. No, this one is broadly applicable. And then also lecture contents will be distributed through the email and then also uploaded to the center YouTube channel after course is complete. And then next week, we will cover ectoderm-derived tissue. What is the ectoderm? Derived tissue, brain, skin, eye, and much more. Lecture 7, neural differentiation in 2D and then 3D system. And then this kind of AI-based image analysis is also a very important combination with bioprinting and then data collection. Even if your research is not focusing on this tissue type, as mentioned, the developmental principles are broadly applicable. In vitro differentiation closely mimicking the embryo differentiation. So development, so understanding the development of biology is essential for the optimizing differentiation across the 3 germ layers, ectoderm, endoderm, and the mesoderm. And this lecture will really hit the core part of this knowledge. And then even if you know much about the development of biology and then stem cell biology, this lecture will summarize what is the main concept. And then we will also have a special guest speaker, Dr. Christina Correa, and from Mayo Clinic, who will present on integrating system biology and the AI in stem cell research and the clinical application. Thanks again for your participation, and we look forward to seeing you next week. Thanks again for Omar, Tom, and Dr. Souza.
Thank
Thanks,
you very
everybody.
much.
Thank you, Kitai.
And I'll share the presentation with you, Kitai, so you can share with the class.
Oh, thank you very much. Just
Yes.
send it to Harry Preston.
Yeah, I will.
Yep.
Yeah.
Thank you very much. Bye.
Thank you.
Thanks, everybody. Have a good week.
Pleasure meeting you, Omar.
You as well.